Hello everyone, welcome back to our lovely series of bio videos. Today I'm talking about cell communication. Alright, so today, as you can see, we got a weird format because it's this way, it's like horizontal instead of vertical. So for those of you at home, which should be all of you taking notes, well you don't have to be at home, but you should be taking notes, make sure you're going this way. Alright, we are going to draw a line like a third of the way down the page. No pretty header today. I don't know if it would fit. Okay, and now that we have this, we're gonna draw like, imagine like there's a huge circle like right here. This is our cell. Okay. So if this is the cell, this will be like the extracellular matrix. And this is the intracellular matrix. So what we're going to be talking about today is how cells will take a signal from another cell and do something about it. So if a cell over here says we need to make a new enzyme, it can send a signal to this cell and say, hey, we need to make a new enzyme, and this cell will make it. So we can have these little signaling molecules called ligands. So this is one type of ligand. We're going to say that this is a protein-based ligand. And we'll also have steroid-based ligands. They're not actually all triangular or circular. This is just how we're going to represent them. But these ligands will be emitted by a cell so that another cell can take on the signal that it wants to pass on. So, in the cell, we have, if you remember from our cell membrane video, we have little proteins embedded in the cell membrane. This one looks like this. They don't all look like this. I actually don't really like, I feel like this doesn't fit. My protein is gonna have a little mouth and then a little tongue, it looks like. Okay, so this is our membrane bound protein receptor. And just to make it pretty, we can color these all in just so you can see them really nicely. So all these ligands, so I'm representing the protein-based ones as triangles and the steroid-based ones as circles. You can also color code those if you want, but I'm just color coding it to make it look nice, I guess. So we'll have a protein receptor in the membrane, and we'll also have protein receptors inside of the cell. So this one, you can kind of tell, is shaped in a way that it'll bind really well with one of these. The thing about our protein-based ligands is we can, this doesn't feel like it's going to fit very well. I'll draw another one right here. So maybe it's like going through the cell, or through the extracellular matrix, because we're not in a cell, we're in like this space in between cells. This is more of a thing in animal cells where the cells aren't like packed super tight, but that's okay. So this protein-based ligand, it can't make it through the cell membrane. This is our plasma membrane, our phospholipid bilayer. It's okay for small molecules that are nonpolar to sometimes diffuse through the membrane, or small molecules that are maybe charged go through ion channels, but a big old protein doesn't just like fuse through the membrane, so that's pretty tricky. But you can have steroids going pretty easily through because they're nonpolar, so we'll have this guy. Imagine, oh, he got smaller. I guess if we're drawing an arrow from that guy, we can draw an arrow toward that guy. Okay, so imagine this guy is traveling into this receptor, and then this guy is traveling into there. So these are both protein receptors, but these signaling molecules, one is a protein, one is a lipid. So this is still a protein receptor, it's just not bound in the membrane. And so this guy can move. This guy is stuck, obviously. So the whole point of this cell communication is we're transmitting a signal to be expressed. So we're transmitting a signal so that we express a gene to make a protein or something. So let's draw a nucleus over here. Not really to scale. Actually did not need to be that big. This is our nucleus. So we're gonna have a lot of stuff to draw, turns out. So there's DNA in here. This does not have to be perfect. We're just saying, is the nucleus that has DNA. Okay. If you want to draw a perfect double helix, you can. You don't have to. And our protein receptor that accepts our steroid-based ligands can just move freely in the cell. So I'm gonna have mine, we're gonna draw some stuff here. So I'm gonna have mine go around on the outside, do a little loop-de-loop, and it'll enter the nucleus, and that's going to express a certain gene. So if this ligand means we make enzyme A, it'll go and basically activate the gene that will eventually make enzyme A. So expresses gene of interest. If you want to learn more about gene expression, that's super important to bio. It's in the bio video playlist linked right here. Not all the videos are by me, but they are all very, very similar. All right, so this protein receptor can travel straight to the nucleus, easy peasy, but this one is stuck. So for this one, we are gonna need a second messenger. So our first messenger is here, and now we're gonna need a second messenger. And one example of a second messenger is cyclic AMP. So to make cyclic AMP, we take ATP, we have lots of ATP in the cell, it should, because that's our huge source of cellular energy, and we can make cyclic AMP, or CAMP. Remember ATP, triphosphate, we lose 2 phosphates and we get monophosphate. And if you want to write down that it's cyclic, you can. Cyclic. And this is our second messenger. So what CAMP, our second messenger, is going to do, I'm going to highlight her because she's pretty important. Second messenger is pretty important too. I guess we're just going to highlight all these things. This is a protein-based ligand, this is a steroid-based ligand, this is a protein receptor, this is a membrane protein receptor. Okay, cool. And this is the nucleus. Let's just highlight everything. Okay, so our second messenger, our CAMP. What CAMP is going to do is it's going to bind to this thing called a protein kinase. And we're going to call this, just as an abbreviation, PK1. And you're like, PK1, is there a PK2? Well, just you wait, there will be. Okay, I'm gonna draw like a little dotted line that says, I'm binding to this. And when it binds to it, it's gonna activate this PK, this protein kinase. So, what a kinase is a type of enzyme that phosphorylates other proteins. Okay, so this is PK1. So what protein kinase PK1 is going to do is it's going to phosphorylate other protein kinases. So now that this one is active, it's going to activate another one. So it's going to use ATP and turn that into oh, well, that doesn't really touch. We liked it to touch. Okay, we're going to turn that to ADP, so adenosine diphosphate, meaning there's one phosphate that's inorganic left over. And it's going to take this inorganic phosphate. I'm actually going to draw this as like a little orb. It's going to take this phosphate and phosphorylate another protein kinase. So, protein kinase, PK2. Okay, from now on, I'm just going to abbreviate as PK, because nobody wants to write protein kinase every time. Okay, so now that we have this inorganic phosphate and we have energy from the ATP, we can turn this PK on. So I'm coloring it green because it's activated. Oh yeah. And we're gonna do that to another PK2. Because now that this one is activated, it can do it to a lot more. And that's going to amplify our signal. So we're spreading it a lot faster because we are spreading it to more enzymes. Okay, so we phosphorylate this PK, it's now active. Guess what's gonna happen? Protein kinase phosphorylates things. So we're gonna get kind of crazy. So more ATP turns into ADP plus an inorganic phosphate. And guess what we're gonna do with that? We're gonna phosphorylate another PK. This time it's PK3. You sensing a pattern? So this one's been phosphorylated, hooray. And so each protein kinase has the ability to phosphorylate multiple other protein kinases or other enzymes, because a kinase is just an enzyme that likes to phosphorylate things. Okay, so we can phosphorylate another PK. And do you really have to draw all these? No, but it's kind of nice to see this, what we call a phosphorylation cascade, where we are seeing this like huge amplification. Every single step is getting larger, phosphorylating more and more enzymes. Pretty cool. Guess what we're gonna do? We're gonna phosphorylate PK3. And then we'll do one more. We're just gonna show each of these doing 2 to show that the signal is being amplified. So we start with 1 PK1 and then 2 PK2s, and then we're gonna have 4 PK3s. ADP. ADP plus inorganic phosphate. Remember that PI is just saying inorganic. The phosphate is not attached to an organic molecule meaning one with carbon. Okay, PK3. So now that these are active, they can do other stuff. And don't worry, we are done with the protein kinases. I bet in biology, generally, this can get pretty extensive, but we're just gonna show these like 3 generations here, which is still plenty. So what's protein kinase gonna do? It's gonna phosphorylate. This one's gonna break down ATP. Don't worry, we're not gonna draw another like level of protein kinases. We are gonna draw this PK phosphorylating something. What's it gonna phosphorylate? Well, this guy is gonna phosphorylate a transcription factor. And what a transcription factor is it's something that now that it's activated, it's gonna serve the same purpose as this intracellular protein receptor and go to the nucleus and say, hey, I wanna activate this gene. Let's transcribe this region of DNA and make a certain protein. So both of these methods are a way to get into the nucleus and say, hey, I want to express a certain gene. What we're gonna do now is we're gonna talk about why would you use a protein-based ligand that has this whole phosphorylation cascade versus a steroid-based ligand that just goes straight to the nucleus. If you wanna say like, this is the phosphorylation cascade, you can. Yippee. Okay, we're gonna break this into about thirds. Do your best. It might get a little bit squishy in here, so we'll see if we can write everything down. Wow, that was not a very straight line, was it? That's okay. All right, so first we're gonna talk about reception. This is our first step. Our first step being we are receiving a signal. Okay, so how does the target cell receive the ligand? Okay, so as we know, there's the protein-based ligand, our little triangles, and this is polar, right? If you remember our macromolecules, proteins are composed of amino acids, which contains various R groups. Some of them are non-polar, but some of them are polar, so a protein will almost always end up being polar, if not always. And since it's polar, it's water soluble, because remember, water is a polar molecule. So it'll be able to mix well in the water because like attracts like. Okay, and this is something that's binding extracellularly, as we notice here. Extracellular binding. And most importantly, why would you do this process versus a steroid-based ligand is this is going to exhibit a short-term response. Okay, well, what about our steroid-based ligands? Okay, we might get a little squishy here, so make sure you're using your space efficiently, because I don't even know if I am. Okay. No, I put an S on that one, I'll put an S on that one. Okay, so these are non-polar, they're fat-soluble. And that's how we're dissolving through the membrane. Obviously, this is binding intracellularly, as we see. It's binding inside the cell. And this is a long-term response. Sorry, you couldn't see. This is a long-term response. So a really good example of this is sex hormones. Also, as you can see, I made a mistake, so we have a little piece of paper covering that up. So an example, you know, your typical one, testosterone. I don't know if estrogen is fitting in here. Estrogen. And also progesterone, but we are not fitting her in there. If you will bear with my barely intact page. Okay, so we have our protein-based ligands and our steroid-based ligands. The protein-based ones are protein and water-soluble, which means that they will bind extracellularly because they can't travel through the membrane. Critically, it's for a short-term response, whereas our steroid-based ligands are a long-term response. And so our nonpolar ones, our steroid-based ones, they are nonpolar, fat-soluble, and they dissolve through the membrane. That's really important. So they bind intracellularly. And some good examples are testosterone and estrogen are sex hormones. Okay, so what is our next step? If our first step was reception, the next one is going to be continuing the signal. It's going to be transduction. Okay, pretend that looks totally normal and like I'm not trying to keep my pages together. Okay, our next step is transduction. Because this is our signal transduction pathway. Oh, you can see my piece of paper. Okay, so how do we get the message to the nucleus, right? So this is going to be very much focusing on our phosphorylation cascade. With our intracellular protein receptor, we go straight to the nucleus. There's no like change happening here. This is more focusing on our protein ligand. So we're looking at our phosphorylation cascade. Okay, so we have our protein kinase. I'm going to draw it as a little Pac-Man. Oh my gosh, pretend that says protein. Protein kinase. Okay, we're going to draw this lovely enzyme. How am I going to draw him? We're going to draw him like this. That's what he looks like. Okay. And if he is phosphorylated, he will look basically the same, but his structure will change a little bit. So maybe he looks like that now. And he'll have a little phosphate group attached somewhere. This is still protein kinase. It just has our lovely phosphate group attached. And so now it's active. So this is our active form. This is our inactive form. And if this guy is dephosphorylated, it'll go back to the inactive form. So once our signal is transduced, it is spread to the nucleus, and let's say we have enough of this enzyme that we wanted to produce, or whatever protein, then we're gonna say, okay, stop it, undo, we don't want any more transcription factors going to the nucleus and making more of this enzyme, we have enough, okay? So we're gonna dephosphorylate them, we're gonna inactivate them. Don't mind my lovely whiteout. Don't actually have whiteout. This is an eyeliner because I'm out of town right now, and that's what I have. Elation. Little squishy in there. Okay, so we can phosphorylate, activate our protein kinase, or dephosphorylate and deactivate our protein kinase, make it inactive. Okay, like I said before, protein kinases, they are the type of protein to phosphorylate something. So we can say kinase here. You can, if you don't have space, you can write it somewhere else. But the kinases are a type of enzyme that phosphorylate. If you don't know, if something ends with ase, it's probably an enzyme. So kinase is an enzyme that phosphorylates things. And there's also a type of enzyme that dephosphorylates things. Ooh, pretend that looks good. And that's called a phosphatase. Which is kind of nice because it has phosph in the name. Okay, so what's happening here is amplification. In our cascade, we're going to have the signal go from 1 to 2 to 4. So each kinase activates more kinases. I'm going to write PKs, which activate more. And this just gets our signal to the nucleus faster. All right, finally, let's talk about response. Although I guess we could go over what we just talked about, do a little bit of highlighting. So our kinases, we'll fill them in. Oh. Okay, so our kinases are phosphorylating, and then, let's use another color. I have a gray marker. Kind of shows inactivation. You don't have to use a gray marker, but a phosphatase is going to dephosphorylate, and that'll be, oh, I should have drawn this one gray. That's inactive. Sad and inactive. And our active one has been phosphorylated by a kinase. But as you can see, they can also be activated by, in this instance, we're having CAMP bind to this PK, and that's turning it on. But I mean, technically there's a phosphate group in there too. So it's kind of getting phosphorylated, but I don't think that you would call it that. Okay, and so we're also just saying we're having this amplification going on in this transduction step where each kinase activates more PKs, which activate more, and that increases our speed. All right, final step response. All right, so what are we doing here? The transcription factor goes to nucleus and we get a response, right? That's the whole point of transmitting the signal. Some sort of thing is happening. So this happens to regulate cell activity. So before I was saying, let's make an enzyme, but all sorts of things can happen. So this usually happens due to a stimulus. So we're gonna respond to something like a change in environment. Some sort of stimulus is going to cause this reaction, and it's going to say, hey, something is different, we need to adapt or something. And this is really important so cells can maintain homeostasis. So if you're not too familiar with the concept of homeostasis, think about like you going outside, right? You go outside and it's 10 degrees Celsius and it is quite chilly. So your body is going to have to do something about it, or else your blood temperature is going to drop and then you'll die, and nobody wants that. So you'll have an outside stimulus like temperature, and that's going to tell your cells, okay, produce more heat, because otherwise cellular systems aren't going to be working anymore, and I don't know, we'll become an ice block. And there's a certain point where you'll like run out of energy or just get too cold, and that's why you don't want to stand outside in the freezing cold for 7 days. You'll not do too well probably. But if you're going outside for a little bit, your body will be able to maintain its normal temperature, exhibit homeostasis. And homeostasis isn't just temperature, it's just like this balanced state that the cells need to survive. So this increases our odds of survival. Basically, what we want to do with homeostasis, that's a key term. Homeostasis. Double highlight. Is we are staying at a set point. We are balancing there. So for humans, that's around 98, 99 degrees Fahrenheit. Don't know how many degrees Celsius, unfortunately, even though Celsius is the real measurement of science, or Kelvin, but who's really talking about Kelvin? Okay, anyway, so here are some examples. Let's look at that. So obviously we were talking about temperature, but there's also gene expression. That's what I was talking about before. We're going to activate a certain gene, make a certain protein, maybe an enzyme. And I say activate here, but what we're talking about is really activating or inactivating. So we are going to be making or stop making a protein. Because that's what our DNA encodes, right? When you learn about the central dogma of biology, our transcription, translation, etc., you're going to learn our DNA contains information to make proteins specifically. So you know how you have carbohydrates and lipids? Those are made in the cell, but when we are expressing our genes, we are making proteins. Because the proteins are the stuff that make the other stuff. But to make the proteins, we got to express a gene in the nucleus. So our gene expression here, perhaps we get a transcription factor that says, I want to express this gene. And so that's going to say, okay, we're going to activate this gene, we're going to make, or maybe we're going to stop making a protein. Okay. What else can be activated by this pathway? What other responses can we see? Well, we can also see cell division. So it'll send a signal that says, okay, we've gone through our checkpoints, we are ready to divide, time to make a new cell. And the next video you're going to watch, if you're checking out my playlist, is not a video by me, but it might as well be. And it's all about the cell cycle, how we are preparing a cell to divide. And then I'm going to teach you about mitosis and meiosis, and that is the process of the cell actually dividing. So we're going to go back and forth. I'm going to teach you, then Miss G is going to teach you, and then I'm going to teach you again. So you'll learn more about cell division later. And then our final example is apoptosis, which is programmed cell death. So there are a couple reasons you'll see apoptosis happening, but basically it's a cell signaling to itself or to another one of its cells to say, hey, you need to stop. It is over. Like something is wrong with you, or maybe we just don't need you anymore. And you need to go. If you remember our cell organelles video, we have these things called lysosomes in our cells, which are full of digestive enzymes. And when we initiate this apoptosis, it'll basically rupture these lysosomes and digestive enzymes will go around and eat the entire cell. So that's one reason we might do apoptosis. Maybe we don't need a certain cell anymore, but we still want the nutrients for the cell. We'll exhibit apoptosis to like recycle those nutrients. Or maybe it's a cell that's dividing like crazy. It's like a cancer cell and we're like, you need you to calm down. Something is wrong. You need to go. Do some apoptosis. Go away. So yeah. Lots of things. So our response is regulating cell activity. This could be gene expression, making a protein, but it can also be something like cell division or apoptosis. It's a reaction to a change in environment, some sort of stimulus that helps maintain homeostasis, staying at the set point. All right. Okay, this is not really working, is it? Okay, little review. Step 1, reception. We have a ligand, maybe it's protein-based, maybe it's steroid-based. The steroid-based ones can go through the phospholipid bilayer. Why is that? Because they're non-polar. They can travel through the phospholipid bilayer made of lipids and go straight to this protein receptor, which is not bound to anything, which means it can go straight to the nucleus. However, these protein-based ligands can't. They'll get stuck if they try to go through the membrane, so they got to bind to a receptor that's also in the membrane, and then that'll initiate this phosphorylation cascade. And remember, this is a more short-term response. The steroid-based ones will be a more long-term response. These are like our sex hormones. So this long phosphorylation cascade includes cyclic AMP, our second messenger. Remember, our ligands are our first messenger. Then we have CAMP, that's our second messenger, and that'll bind to a protein kinase, which will activate a bunch of other protein kinases and eventually phosphorylate a transcription factor, which will go to the nucleus and say, we want to do this now. And transcribe a gene, for example, do some gene expression. So yeah, oh yeah, if you want to dephosphorylate, you'll use a phosphatase. Maybe when this is all over, we have enough of whatever enzyme. And this is also expressing a gene of interest in this example. Okay, well anyway, hope you enjoyed our signal transduction pathway. Like I said, I don't know where you would put the header. Maybe you can write it right here. I feel like you can do it. I will let you make that decision. Maybe headers aren't as important to you as they are to me. I love a header, but you know, with a sideways page, it's like, are you gonna put it this way? Are you gonna put it that way? Anyway, thank you for tuning in, and tune in for the next video on the playlist. Once again, not by me, so don't search my channel, but do search my playlist to learn more about the cell cycle. And then I'll teach you about mitosis and meiosis. Okay, bye!